Welcome to the Raaonline lecture series on renal physiology. In the last session, we saw about the first step in the urine formation, that is the glomerular filtration. Now we will move on to the next step, that is what happens to the filtrate that is produced in the glomerulus when it goes to the renal tubules. So let's see in detail about all these functions. The specific learning objectives of this session would be, we will be talking about the general principles of the renal tubular functions, how the solutes and the water are getting transported, what are the principles governing these transport. Then we will go to discuss about the tubular events in each segment of the nephron, say proximal tubules, loop of Henle, distal tubules and the collecting ducts. Finally, we will see about the regulation of the tubular function. So this part will be dealt in detail pertaining to each and every mechanism of solute reabsorption or secretion, whatever it may be. Fine. Now let us see the structure of the nephron across which the solutes are getting transported. Okay. So this is the tubular lumen. Okay. So what is this? This is the tubular lumen. Before that, I would like to draw the glomerulus here. Maybe this is the glomerulus and you have got the tuft of capillaries here, okay, and from the capillaries the fluid gets filtered into the Bowman space and this fluid now is going to the renal tubules, okay. And when it crosses the renal tubule, you know the structure of the nephron. After the Bowman space, it continues as the proximal convoluted tubule. Then it continues as the loop of Henle, which has got the descending loop, ascending loop. Then it forms the distal convoluted tubule, and the distal convoluted tubule ends into the collecting ducts cortical and the medullary collecting ducts drain into the pelvis, okay, and that is where the urine is collected and from there it is sent out to the ureter. This is the structure that I hope you all know about this. Now when this fluid, it is going into the tubular lumen, there are cells, these are the epithelial cells. You can see this is the epithelial cells, renal epithelial cells, which is called as tubular epithelial cells. These cells, they have lot of functions. Some of the fluid or the solutes, they get filtered into the epithelial cells and from there it gets into the peritubular capillary, okay. Some of the cells or some of the solutes, they get secreted from the capillary into the lumen. This is what is happening. Now, if you look at the epithelial cells, you can see there are tight junctions here. In between the two cells, you have got tight junctions. They are tightly bound to each other. They do not allow the solutes or larger particles to a greater extent, but still there occurs some transport across the tight junctions also, okay. And if you can see here, this is the interstitial space, all these blue color thing is the interstitium, renal interstitium, or which is filled with the interstitial fluid, okay. But in between the cells, there are some spaces, okay. So these are the lateral interstitial spaces or intercellular spaces, right. This is the lateral surface and this is the basal surface, okay. You can see this is the apical surface. So apical, lateral, basal surface. Basal and lateral together we call it as basolateral membrane, okay. And apical membrane is also called as luminal membrane. Why I am stressing upon all these things is, lot of transport mechanisms are happening on the apical membrane as well as the basolateral membrane. So if you can see here, there is space beyond this basal membrane, but there is much more space in the lateral surface. That is very, very important for the transport of solutes, which is actually guiding or giving the driving force for the solutes, okay. We will be seeing all these things in detail in further slides.